Good morning, good afternoon. Welcome to the webinar. My name is David Myatt. I lead communications for industrial microbiology healthcare business at bioMérieux in the Asia Pacific region. We're very pleased that you've joined us today and we'll hope you'll find this webinar to be relevant, interesting and valuable. bioMérieux has an overarching mission of pioneering diagnostics in support of public health. In industrial microbiology, we aim to deliver value by helping our customers with improving product health and safety, business productivity, and protecting their brand reputation. Our iMicro webinar program aims to deliver valuable and timely information, education, and updates on a variety of topics relevant to microbiology practice for the quality and safety of pharmaceutical and healthcare products. Today, our agenda begins with my introduction. Then we move on to keynote presentations from Dr. Chakameh Azimpour from Amgen and Jeremy Robertson from bioMérieux Industry in the United States. Following that, we've got the opportunity to have a live Q&A session. However, we won't be taking verbal questions today. Okay, so let's begin with an introduction of our speakers. The first speaker today is Dr. Chakameh Azimpour from Amgen. Chakameh joined Amgen in 2019 to lead the biosafety development team in charge of mycoplasma testing and viral clearance assessment studies. She's now responsible for driving innovation and technology development efforts related to next generation virus production and purification, as well as cutting edge method and assay development for detection of adventitious agents. Chakameh has been a lecturer and mentor to university students in the fields of microbiology and biotechnology and volunteers in local middle schools supporting STEM initiatives. She holds a PhD in genetics from the University of Vienna in Austria. Our second speaker today will be Jeremy Robertson. Jeremy is Global Solutions Manager for BioFire in the bioMérieux Industrial Microbiology Unit. He's based in Chicago in the United States. Jeremy is a professional microbiologist with more than 17 years of experience in industrial microbiology applications related to analytical equipment in pharmaceuticals, the home and beauty industry, and food and beverage industries. Jeremy has was previously leading and steering the global microbial identification solution business for bioMérieux with great success. He holds a bachelor's degree in biology from the University of Southern Florida. So we welcome both Chakameh and Jeremy as our guest speakers today. Today's topic is about mycoplasma testing, an automated alternative to traditional NAT methods. The standard test for mycoplasma contamination of pharmaceuticals is evolving, as is the need for faster and simpler tests. As macromolecule drugs derived from mammalian cells or other bioprocesses become more commonplace, reducing the turnaround time for testing and investigation on the large-scale bioreactors of today has become an essential need for manufacturers to ensure productivity and cost competitiveness. The pressure to reduce turnaround time has spurred innovation with real-time PCR methods. The vision for future testing is simple to use, fast and accurate. So today we're going to learn more about how Amgen has boosted productivity by adopting a next generation nucleic acid amplification test, an NAT test for mycoplasma, and then we're going to hear something of the technology itself from Jeremy Robertson. So without further delay, I'll hand over to Chakameh. Chakameh, you're very welcome. Thank you for joining us today from California. We know it's getting late there. Thank you very much for joining us.
Hi, thank you so much for giving me the opportunity to talk about the rapid mycoplasma detection method. I actually believe that it would be best if Jeremy goes first and introduce the BioFire system, and then I'll pick up and I'll go through with what have we been doing at Amgen.
Yes, let's do that, Chakameh.
Thank you.
Okay, so greetings everyone, and good morning, good evening, depending on where in the world you are. I want to start by speaking to you today about our BioFire mycoplasma solution, introducing you to our system. Really, our system is the easiest mycoplasma test on the market today, providing detection of mycoplasma in just under an hour. So our system allows easy, rapid at-line testing that can be performed by virtually anyone to test for mycoplasma anywhere at any time. It's a flexible on-demand test that can be used on raw and in-process materials, as well as a release test. Especially today, with short shelf life products and consolidated supply chains, the need to confirm the absence of mycoplasma as quickly and efficiently as possible has a huge value. The BioFire mycoplasma test can be used for in-process controls, as I mentioned. Because no PCR skills or lab are required, quick, simple, and accurate in-process testing can be moved out of the lab and closer to production. Since there are just 2 minutes of hands-on time and a total run time of 46 minutes, testing can be performed on demand anytime there is a need to screen for mycoplasma contamination, which makes it a perfect choice for in-process control testing in bioproduction. Rapid and efficient in-process mycoplasma testing allows you to have an early contamination alert so you can protect your manufacturing process. And BioFire is not just for in-process controls. It's been validated to fulfill the requirements of pharmacopoeias for product release and is able to detect over 130 types of mycoplasma. And because it's an automated test with 21 CFR Part 11 capable software, it can improve your data integrity position as well. As a comprehensive and validated test, BioFire meets regulatory requirements for mycoplasma testing. The BioFire solution actually consists of 2 components, the film array instrument and the single-use consumable that we like to call a molecular lab in a pouch. So with just these 2 items, really you have everything that you need for fast, accurate, and simple mycoplasma testing. The film array instrument is about the size of a small kitchen appliance. And despite the small footprint, the system is able to perform the extraction, amplification, and detection that typically require several larger instruments. Here are some photos of the actual machine and reagents. The standard system comes with 2 units for increased throughput as well as redundancy. In fact, up to 8 units can be added to a single computer if higher throughput is needed. The disposables are packed 6 to a box and are stable at room temperature. So now let's talk a little bit more about these disposables and what's happening in the pouch. As unique as the film array instrument is, the science happens in the single-use disposable pouch. About the size of an index card or a smartphone, the pouch contains all of the reagents required to run the extractions and multiple types of PCR reactions. These reactions and detection occur in the fluidic circuit at the bottom of the pouch, as you can see highlighted here. I'll now go into a little bit more detail about each one of these steps. So after the sample and rehydration buffer are injected, the extraction process begins. Silica and magnetic beads are used for the nucleic acid extraction and purification. After the purification step, The first stage PCR includes reverse transcription with a multiplex PCR. Because we're utilizing both RNA and DNA, presence of live mycoplasma in the sample enhances the sensitivity of detection. The second stage of PCR is a nested PCR. Each well of the array is a single PCR reaction, and we use a total of 14 unique PCR reactions targeting 3 different genes to detect more than 130 species of mycoplasma. There are several controls integrated into the pouch to ensure quality of results. As you can see here, the entire process is monitored from extractions through PCR2 with a freeze-dried yeast. Yeast RNA is used to check the reverse transcription as well in PCR1. And finally, PCR2 is checked by DNA fragment controls. And for additional controls, such as those that are needed for incoming quality control, training, or qualifications, we've developed an external positive control kit that utilizes synthetic modified DNA mix. The modifications shift the temperature and the melt curves so they can be distinguished from natural mycoplasma DNA. Now that we've covered how everything works, let's talk through the workflow. Today, many biopharma companies are using conventional PCR because they realize the benefits of getting faster results compared to compendial methods that require 28 days or even longer for mycoplasma results. Still, conventional PCR requires a dedicated lab, highly trained analysts, extensive hands-on time, and manual result interpretations. And we know there is a risk of contamination because it's an open process, and the manual nature of the test also means that the potential for pipetting variability or human errors exist as well. By contrast, BioFire offers a sample-to-answer PCR method that is fully automated, delivering not only simplicity and ease of use, but also speed, with only 2 minutes of hands-on time, and results in little less than an hour. So now I'd like to show you a brief video that demonstrates how to prepare samples for testing on the BioFire system.
The science happens in the single-use consumable we call a molecular lab in a pouch. The pouch contains all of the reagents required to run the extractions and multiple types of PCR reactions. Preparing the sample is simple. The technician loads the pouch into the loading block. Hydration solution is injected into the pouch through the blue inlet port. The sample is added to the sample injection vial using the transfer pipette. Sample buffer is added to the sample injection vial. The lid of the sample injection vial is closed, and the vial is inverted 3 times to mix the sample. The sample buffer mixture is injected into the pouch through the red inlet port. The pouch can now be loaded into the film array instrument. To learn how anyone can perform mycoplasma testing anywhere at any time, contact your bioMérieux representative.
Okay, thank you, Vivian, for helping me with that video. So, in fact, we've developed 2 protocols for testing on BioFire. For quick in-process control tests, you would simply perform the pouch setup process that you just saw in the animation with 200 microliters of your sample. We validated this protocol with a limit of detection, or LOD, of 33 CFU per mil. To achieve the LOD of 10 CFU per mil that's required for product release, there's a little bit more hands-on due to the addition of a centrifugation step of a 10 mil sample, as you can see here on the screen. But even with the centrifugation steps needed for higher sensitivity, the total time to result is still less than an hour and a half compared to several hours or even days with traditional PCR or compendial methods. Once the pouch is set up, you can start the run using our intuitive software. Despite the simplicity of the software, it's been specifically developed to meet pharma standards. Features like barcode scanning and automation help reduce the risk of human error and increase data integrity, while a suite of features such as audit trail and user logins help allow compliance with 21 CFR Part 11 requirements. And like everything else with the BioFire system, result reports are also simple and straightforward. You can see here in these example reports that the results are simply mycoplasma detected or mycoplasma not detected. Additionally, the reports indicate whether or not the internal controls passed. So now let's take a look at how we performed our internal validation of the BioFire system for mycoplasma testing. We've used international pharmacopoeia guidelines to validate the BioFire method, using the criteria described in the major chapters for mycoplasma testing, such as LOD and organism type. Similarly, we have used the various chapters describing validation of alternative methods for qualitative tests like BioFire, and these require the parameters of specificity, limit of detection, robustness, repeatability, and ruggedness. I'm not going to go into detail on these data, but we do include the detailed primary validation data in our validation report that we provide to users as part of our validation package. The validation package also includes an installation, operational, and performance qualification template that you can utilize for system qualification as well. So to summarize what I've presented to you today, we can compare the next generation nucleic acid technique BioFire test to conventional PCR methods as well as compendial methods for detection of mycoplasma. As you can see, while BioFire is able to achieve the sensitivity needed as a release test, it does so as an easy-to-use sample-to-result system, providing a qualitative presence-absence result in just under an hour. With low contamination risk, easy reagent storage, and a small footprint, the test can be performed outside of the lab compared to other methods that require specialized equipment and a significant amount of training. All of these features allow mycoplasma testing closer to manufacturing so that the test can be conducted on demand whenever confirmation of the absence of mycoplasma is needed. With rapid results that can be obtained anywhere at any time with BioFire, you can get an earlier alert to contamination compared to other methods. This can help minimize the impact to production when a contamination does occur. As a load and go system, BioFire allows users to reallocate resources to other tasks, thereby increasing their productivity. And our comprehensive validation that we mentioned earlier meets international regulatory requirements. So I think that you can see from this presentation, I hope you can see the simplicity of our approach. There are a number of very real benefits to that simplicity, including the reduction of labor costs, reduced risk of human error, increased testing flexibility, and the ability to minimize the impact of a contamination event if and when it does occur. So with that, I will thank you very much for your attention, and I'm going to hand it over to Chakameh, hand it back to Chakameh who's going to present to you a real-life example of BioFire mycoplasma system usage. Thanks, everybody. All yours, Chakameh.
Thank you, Jeremy. I just want to make sure you can see my screen. Does it show properly?
Looks good.
So yes. So thank you again, Jeremy, for the introduction, and thank you for giving me the opportunity to talk about the rapid mycoplasma detection method that we tried at Amgen for use in detection of mycoplasma in biologics and cell therapy products with short shelf life. I'm just going very quickly to review the agenda. I thought it would be a good idea if I talk about the key study drivers. I will briefly take a look at the regulatory guidance for developing mycoplasma testing, even though Jeremy also mentioned that. I will talk about the challenges that we had in mind for the rapid microbiological detection and the need for the rapid testing, and very, very quickly touch base about our reason to try the BioFire method. I know Jeremy just covered the BioFire system in details, but I wanted to tell you what is our perspective as an end user on the topic. And then I'll share the rapid mycoplasma testing that we tried, and I will share the data with you. I hope that it would be useful for you all. So before I start, I think it's important to take a look at the cost of mycoplasma contamination and see why is it so important to test it. As you know, mycoplasma are a type of bacteria containing very unique property that making them very challenging to both remove and detect in cell culture. Compared to many bacteria, mycoplasma are very, very small, and they can pass through the commonly used sterilizing grade filters that we use to prepare the cell culture media components. And once the cell culture is contaminated with mycoplasma, because of their small size and the slow growth rate and the minimal impact that they have on the turbidity, it's very difficult to detect them through the optical microscopic evaluation and the usual turbidity measurements. So depending on where or when mycoplasma was introduced to the bioprocesses and the quantity that they are introduced at or the specific species, it can affect the host cell performance that will cascade to many consequences. So no matter which one of the mycoplasma is the causative agent, or no matter how they've been introduced, they will cause product loss, actually a huge product loss. And when they are detected, all the processes must be halted, the product in question must be disposed of, we have to start the root cause analysis, and it can even potentially result in the plant shutdown if they're present in the final drug product, which is a significant economic loss. In addition, some mycoplasma, like Mycoplasma pneumonia, is a human pathogen and will directly put the health of the patients at risk. And so looking at this, you can imagine that the cost of mycoplasma contamination isn't something that can easily be tolerated. So to minimize this risk, we have to have routine testing for mycoplasma that's performed throughout the product manufacturing and developmental processes. Ensuring the biosafety of the cellular preparation is presenting an ongoing challenge for manufacturers, for regulators, for physicians and patients alike. These common contaminants like mycoplasma, bacteria, fungi, and viruses are currently detected by compendial method as the gold standard. But as you know, and as you've heard Jeremy mention it, they're time-consuming and they're not the best fit if you're trying to develop a methodology for a short-life cell product. Especially for autologous cell therapy manufacturing, we really have to find a different control strategy to maximize on the opportunity for therapeutic benefits to the patient while maintaining their safety. Also, as you probably know, there is only very small amount of sample volume available for testing in this type of manufacturing. So here at Amgen, we are leveraging a very well-established control strategy that we have when we are developing these rapid testings. Now, with that in mind, let's look at this complexity of different guidelines that one have to consider when developing a method for mycoplasma detection. Thankfully, in the last year, FDA published a new guidance for industry that explains the CMC information for human gene therapy INDs. Combining this with already existing documents covering the US pharmacopoeia, FDA guidelines, and point to consider the European pharmacopoeia and the Japanese pharmacopoeia for mycoplasma testing, in addition to the guidance on the validation of alternative microbiological methods. We very carefully follow these guidelines that explain the expectations on testing adventitious agents, including mycoplasma for cell therapy products and also CHO-based samples alike. So having the guideline in mind, let's take a quick look at the generic view on the cell therapy workflow. Here in this picture, you can see a generic patient-specific adaptive immunotherapy workflow. As you can see, the donor leukapheresis samples, they undergo selection to be enriched for the cell of interest, for example, T cells or NK cells. Then previously manufactured viral vectors are added to genetically modify the immune cells, conferring new cancer recognition receptors. These modified cells will be then expanded to therapeutically relevant number, followed by the downstream processes, including washing, formulation, bagging, and cryostorage into an infusible patient dose. Now, before the final cell product is shipped to the clinical site where it's thawed and then administered to the patient, they have to be tested for microbial testing, including mycoplasma. And as you can imagine, a compendial method that might take 28 days won't be suitable. And plus, as we mentioned, there is really a limitation on the sample volume to consider as well. And also something to remember, it's the patient-to-patient variability. So faster turnaround times and these type of processes for testing will enable a real-time in-process sample testing. Also, another important benefit is that after the initial qualification is completed and you have a method in hand in-house, these type of in-house testing are very cost-effective and provide a very good control over investigations if there was any contamination incidents to happen, and therefore you can adjust the testing strategies very, very quickly if needed. Now, with regulations and a brief overview in mind, I tried to summarize the available mycoplasma testing or the current available mycoplasma testing in one table. So I listed the compendial method, the Amgen modified lot release method, also the qPCR, and the BioFire. If I start with the compendial method, you probably know, or I'll just go over it. They involve growth of the cultivable strain in the liquid broth or on solid agar, and they're capable of detecting 10 colony-forming units per milliliter of samples. For the non-cultivable samples, we have to use mammalian cell cultures like the Vero. But regardless, the growth is detected by staining the cell cultures with the fluorescent dye-binding dye, followed by visualization by microscopy. Now, these conventional methods, they provide very effective mycoplasma detection. However, they're very, very time consuming, and a minimum of 28 days is required to perform. This lengthy turnaround time can be very problematic in the field of cell therapy, and especially if we're working with the non-cryopreserved cell products that they expire very quickly within 24 to 48 hours. And also in those occasions, you need the mycoplasma test results immediately to inform for treatment decisions. So we definitely need a shorter time period. Amgen's adopted lot release testing, which is an improved method. It takes only 8 days and it's regulatory approved. But for cell therapy product, we're interested in developing even more rapid testing. The qPCR testing or PCR testing, as Jeremy also mentioned, they are a potential solution for the issue of time and also for the sensitivity, because they detect the presence of nucleic acid sequences, which are very unique to that potential contaminant. And they're very sensitive and they can very rapidly be executed. But you still face the challenges of the setup. You have to train the staff. And in addition, for every one of them, you have to validate it in-house. You have to meet regulatory requirements when developing a new assay. And then we came across the information for the BioFire, which was interesting to us because the promises of the fast time to results. Although, again, similar to the qPCR, we have to go through the validation of this process. But regardless, we were interested in the beginning by, as I mentioned, the promise of the fast time to results. In addition, it was a positive point for us that it covers the range of mycoplasma species that are required for regulatory agencies, including the Japanese pharmacopoeia. And we liked it that it's a closed system that lowers the risk of contamination, and it offers a very easy to generate reports compliant with 21 CFR Part 11. As Jeremy mentioned, it's a very small and very easy to use pouch, and the equipment is easy to perform. Our staff could easily be trained and they perform the testing. Now, the bioMérieux, as Jeremy explained, has 2 approved protocols for direct testing with 200 microliter of sample or a 10 milliliter test sample similar to compendial method requirements that can go through 2-step centrifugation. I'm not going through explaining the differences between 200 and 10 milliliter because Jeremy nicely covered them all. But I wanted to add that at Amgen, internally, we decided that in addition to the 200 microliter testing and the 10 milliliter testing, we also wanted to test an additional volume, and then we tested 1 milliliter of samples spiked with mycoplasma. For the 1 milliliter, we followed the very same protocol as the 10 milliliter, meaning we started with that gentle centrifugation, collecting the supernatant, going through pelleting the mycoplasma with higher speed centrifugation, and then we followed the protocol exactly as it would be for the 10 milliliter. So in summary, we tested at 200 microliter, 1 milliliter, and then 10 milliliter. So another thing I want to emphasize is that for this testing, we chose the CHO-based test samples because we had the wealth of information available for them to be able to compare our results. And we also chose to do this study on our most challenging sample, and we tested these samples with mycoplasma spike at the very, very low CFU per milliliter of test sample. I want to mention that we tested the 200 microliter, again, 1 milliliter and 10 milliliter. And for the 200 microliter, as Jeremy mentioned, the LOD that it's claimed by the vendor, it's less than or equal to 33 CFU per milliliter. Now, what we've done is that let's consider sample 1. It was a spike with 3 different types of mycoplasma. We tested mycoplasma pneumonia, mycoplasma hyohines, and mycoplasma orale at the spike levels that it's mentioned here in the table. And we tested each one of these samples in triplicates. Means, so sample 1, spiked with mycoplasma pneumonia, tested 3 times at 200 microliter, 3 times at 1 milliliter, and 3 times at 10 milliliter. As you can see in the table, at the 1 milliliter and at the 10 milliliter, we covered 100% detection for all three spike samples. For mycoplasma pneumonia, we only got detection at 4 out of 6 tested samples, which is the 67%. You might think that 67% is not a very good result, but in our view, because of that low spike level, we were excited to get these samples, and therefore we moved on with the second sample. We spiked it with mycoplasma gallisepticum, spiked at 2.7 CFU per milliliter, and we still received 100% detection across the board with triplicates of at 200 microliter, at 1 milliliter, and also at 10 milliliter. Moving on, we tested sample 3, spiked with mycoplasma pneumonia and mycoplasma gallisepticum. Very similar to what we had before. For mycoplasma pneumonia at the 200 microliter testing, we did not get the 100% detection, but at 1 milliliter and 10 milliliter sample, 100% detection was obtained. Also for mycoplasma gallisepticum, 100% across the board. And sample 4, spiked with mycoplasma hyorinus and mycoplasma orale, also performed 100% regardless of what sample volume they were tested at. And so for us, it was a positive looking results. It seems that the BioFire system consistently is able to detect the spiked mycoplasma even at the low CFU per milliliter in derived cell culture. So the two main challenges that we were looking into, they were positively responded. A small sample volume could yield positive results, and also we had a significant improvement in the testing. Because of these positive results, we were interested in evaluating additional application of such rapid testing, and therefore we are looking into further applications. We are interested in evaluating the BioFire for risk mitigations through the process, or maybe for the release testing of the derived products. But overall, at least for the part of the study that we tested, we received quite positive outcome. And with that, I come to the end of my presentation. Thank you again for being here, and I'm ready to answer any question that you might have.
Thank you, Chakameh. Really very interesting, very impressive. Thank you for that presentation and the insight into what you've done at Amgen to bring this solution into your workflow. Thank you to Jeremy for the insight into the platform itself. We've got a couple of questions that have come in, so let me take this opportunity to invite the audience to offer up any questions you've got in response to both Chakameh's presentation of how they've gone forward with this at Amgen, but also in relation to the technology itself as it was presented by Jeremy. So feel free to put your questions in the webinar interface, which appears on your computer screen. As I said before, there's a little grey box with the word questions and a little white arrow to the left of the word questions. If you click on that, it will open up a text box where you can write a question. Or if you're on a mobile device, in the grey margin at the top of the screen, you'll see a question mark. If you just touch that question mark again, it will open up a text box where you can offer up a question. So while people are giving that a moment's thought, feel free to go ahead and ask your questions, but we do have a couple that have come in. Jeremy, maybe you'd like to turn your camera on as well, so the audience can see all of us. And let's start with a question about the potential for contamination here. One of the questions that's come in from Korea is a detailed question about whether there's any potential for contamination of the PCR process, because like DNA contamination of PCR processes is a kind of well-known phenomenon. And how did you deal with that in terms of the design of the experimentation, the validation work that you've done? Chakameh, can you speak to that topic at all?
Is it about the development of the BioFire pouch, contamination in that PCR, or contamination in the PCR testing as like a regular release testing for mycoplasma for the in-process testing?
Well, perhaps from both dimensions. Did the BioFire pouch actually offer you any advantage over risk of contamination in the transition that you've made from a more traditional approach to a rapid approach?
So for contamination for the in-process testing, I have to say this is something you don't want it to happen, right? So there are a very rigorous contamination control strategy in place. And therefore, these PCRs for risk mitigations are only as risk mitigation as their name proposes. So at various steps within the manufacturing processes, a PCR will be performed. It's not being performed by us. Usually it's performed by the QC departments in biopharmaceutical industry. And also there are multiple controls, as like an extraction control or just the PCR control within the system that helps ensuring that your PCR is performing as intended. I hope I answered the question, if I understood that right.
Yeah, no, I think you have. And I guess it's kind of a related concept. What about the potential for interference? I guess you would have had to design into the validation process the
possibility of the interference. What we do is that we usually test the derived testing with known mycoplasma before we perform the release testing, and therefore we know whether there is an interference in the sample matrix that would prevent the PCR assay or not.
Great. Okay, thank you. We've got a question come in from, actually I'm not sure where it's from, but it's directed specifically to Jeremy. Jeremy, what's the status of the BioFire mycoplasma testing panel CMC filing and approval in the US? I guess that's related to our activities. And if I can add on to that, are there actually any FDA approved products on the market that are using BioFire mycoplasma?
Yeah, good question. Important to remember, the FDA will approve an application as submitted with the test protocol that is submitted in that application. So we don't have the BioFire is not per se an approved test, but we follow the guidelines and the recommendations we have the capabilities to be approved. It's designed in that respect. It has been approved as a 510K clearance. That's more on the clinical side of the business, of course. They are using this same system on the clinical side to test for a variety of different things, not just mycoplasma. We don't currently have a customer who is approved to use the BioFire from FDA, but there are 2 customers currently who are pursuing that approval, collecting validation data and pursuing that approval in the coming years. It takes some time, as you know, to collect the data that's necessary. And the system is still relatively new on the market. So we expect to have those approvals soon, hopefully.
Okay, another question has come in related to the sensitivity. So this is possibly, I think this relates to the stuff that you said, Jeremy, about the two different protocols. The question relates to the European Pharmacopoeia section 2.6.7, where it's recommended that nucleic acid amplification tests that are intended to replace the compendial culture method, the LOD needs to be proven to be less than or equal to 10 CFU per mil. But the mention of BioFire having a limit of detection of 33 CFU per mil, how was it justified that BioFire is sensitive enough to replace the compendial culture method?
So yes, so we have the two protocols that I outlined in the presentation. The 200 microliter protocol indeed has the LOD of 33 CFU per mil, but we also have that 10 mil protocol, which gets us up to that higher sensitivity of 10 CFU per mil.
Okay, thank you. Now, there's two questions here that have come in on a similar sort of topic, and maybe Chakameh, you're able to comment on this, but maybe Jeremy can chime in as well. This relates to the attachment or the association of mycoplasmas with the actual mammalian cells, and these protocols involve centrifugation and the potential loss of mycoplasma organisms through the centrifugation process, or is there a risk of reduced sensitivity in situations where the cells are being lysed or the cell components are lost due to centrifugation?
Well, in our case, what I can say from our side is that the first centrifugation, it's at a very, very, very low speed. It's a very gentle centrifugation, and the chances for loss of mycoplasma, it's very minimal. And we have controls in place. Therefore, I wouldn't take it as a risk, but I also would leave it to Jeremy to say it from their approved method for the 10 milliliter.
Jeremy, have you got a comment?
Yeah, just I just want to reiterate the two different protocols that we have. The 200 microliter protocol that does not require centrifugation does not exclude cellular material from the sample as well. So I would just add that point as well.
Okay, thank you. We also have a question about that relates to the usual issue, if you like, of excuse me, I need to get rid of that. That relates to the usual problem of sensitivity and specificity, or the usual, not problem, the usual questions about sensitivity and specificity. And again, maybe both of you can comment on this, starting with Chakameh, within your experience now with the BioFire mycoplasma test. Have you seen have you had any experience with false positives? And Jeremy, I would ask if you're aware of a false positive rate, or whether there's a claimed false positive rate for BioFire, and then the sensitivity of the system. I think Chakameh, a little bit you talked about that in terms of your validation project, but have you got a broader comment to make on sensitivity as well?
Well, I can, again, sorry, Jeremy, I chime in from our end user perspective. We actually didn't see any false positive in all the on-site samples that we tested. So we haven't seen it, but we also tested a limited number of pouches. So probably if there are more data available, Jeremy can mention that. And also for the sensitivity, as you may have noticed in our data, we challenged the system at a very low spike level. So the detection level was, we're looking for 10 CFU per milliliter, but we spiked at a very low level. And across the board for 1 milliliter and 10 milliliter, we were able to detect them. So sensitivity in our view for the samples that we tested was acceptable, and we didn't see any false positive.
Yeah, and I would add that our internal data shows a false positive rate of less than 1%. However, that data was collected under very controlled conditions, and as we've designed this system to be used outside of the lab and really anywhere, I would expect perhaps we might see a slightly higher rate of false positives once the system is in use in the field. But we have a limited number of data set to base a conclusion on, I think, at this point. But so far, what we've seen is in the range of what our internal studies showed as well.
Okay, thank you for that. Another question about interference with the, this is not so much about contamination, but about interference in the chemistry of the PCR test in the pouch that might arise from media components, those sort of things, particularly in the context of the in-process testing, or indeed if you were using the test for testing finished goods, fully formulated pharmaceuticals. Is there any potential for interference in the test from components of the formulated product? I guess you've given some
Well, to my understanding, the pouches, they have the internal control from the yeast DNA, as Jeremy explained. And so if they don't pass, it shows that there is interference. Therefore, one of the internal controls, if they fail, then you don't get the system suitability, and therefore you're not going to get results for your mycoplasma detection. So the interference and inhibition should be checked and get the green light from the system. But that's my understanding. So Jeremy, if there is more into it, please feel free to.
In fact, in fact, I'm going to pull in our technical specialist, Nadia Ward, who I think can address that question perhaps a little better than I can. Nadia, do you want to add anything to what Chakameh said?
Certainly, yes. I would like to add that during our primary validation of the BioFire technology and pouches, we did assess a variety of different interfering substances. And these substances included seras, different cell lines, microorganisms, disinfectants, cryoprotectants, antimicrobials, anything basically that might be added to the manufacturing process. And we've shown that there's really nothing so far that has demonstrated a major interference with the performance of the pouch. The only disrupting substance was 1% bleach, but this is actually something that we expect. In addition to that, and I was just trying to get a better understanding of the question, as Chakameh said, the 3 internal controls that are integrated within the pouch are extremely important, and they will give you a clear indication if there is anything that is interfering with the PCR processes. So it's important to pay attention to these, and in fact, should any of these internal controls fail, the entire assay will be considered invalid and we would not report out a result. So that being said, this assay from a technical standpoint is quite robust, and even known PCR inhibitors, we've seen some customers test products such as sodium heparin at different levels and have not found that to interfere with the PCR processes within the pouch. Thank you.
Thanks, Nadia. That's a good comment. I guess it's related. Someone has also asked about whether high calcium containing samples need to be diluted, and indeed what matrices have actually been validated so far that we're aware of?
You know, I'm not sure about the high calcium specifically. I don't know if Nadia, you have any comments on that, but I can say that we have tested a variety of different materials on the BioFire system and have not found interference from them, aside from the bleach that Nadia mentioned. That information we have published in a technical poster that we presented at a trade show last year, but so it's available. But yeah, we haven't really found a whole lot that has interfered with the test so far. Nadia, do you have anything to add?
No, I was going to agree with that. And in addition to the interfering substances list that we've published, we've also done more than 12, 15 feasibility studies in our R&D labs across the world. And those clients have provided us materials with different matrices and have provided us some information about the formulation. So if there is a specific component that is of concern to you, we could cross-reference this with our internal library of matrices that we've already assessed.
Thanks, Jeremy. Thanks, Nadia. Okay, so we have just one last question at this point. We have a question relating to the maximum number of human cells or mammalian cells that can be introduced to the test.
Our protocols today function best at less than 10 to the 6, but we are working on some additional protocols to be able to test higher cell density samples as well. Thanks,
Jeremy. Okay, so we're past the hour at this point. So I'd like to draw the Q&A session to a close. I want to point out to our audience that both Jeremy and Chakameh, and for that matter Nadia, are all located in the United States. And therefore it's rather late in the evening. So I really do want to thank you all for joining us at a suitable time for the Asia Pacific time zones and for staying up late to present to us today. It is really very much appreciated. Thank you very much.
Absolutely, it was our pleasure.
My pleasure, yes.
So thank you, and I also thank our audience for joining us today. I think we had about 70 people in the audience today across the Asia Pacific region. So thank you very much. And with that, I wish you all a very good afternoon and a good evening in the US. And we look forward to seeing you again soon.
Thank you. Bye-bye.
Thank you.
Thank you.
Bye.
Thank you. Bye-bye.
Have a great day.